My name is Olivier Binner, and I'm a postdoctoral researcher at the MRC Mitochondrial Biology Unit, which is part of the University of Cambridge. And I'm going to talk today about how to build synthetic cells from the bottom up. So most sciences traditionally use a top-down approach, which is you start with a very complex problems and you try to decompose it into simpler and smaller problems. And then by answering the simple problems, you will learn something about your complex problem. And this is the traditional approach used in subjects such as biology, physics, and astronomy. However, it is quite limited because you can only learn what is already there. So you can only understand what is already there. For instance, when we look at an organism, you can look at its evolution, how its biochemistry is working, at its cell biology, genetics, physiology, and anatomy, and we will learn a whole lot about the organism. However, this approach cannot create a new organism with new functionalities. And this is a bottom-up approach, which is normally used in chemistry and engineering. For instance, an engineer can create a beautiful watch from all these individual parts. It's a very complex machine and device. Or a chemist can synthesize a very complex molecule in a multi-step synthesis. And one of the biggest questions in science nowadays is if this bottom-up approach can be applied to biology. So if we can use all the knowledge we gain from these individual subdisciplines to create something like a new organism. So if we can apply chemistry to build something new in biology. And the ultimate goal in this research is to create a living system from the bottom up in a test tube. And this means that we try to find the minimal set of molecules needed to convert non-living matter into living matter, which equals the creation of a synthetic cell. And this is a very interdisciplinary research field, which involves principles from chemistry, biology, physics, engineering. And once you create synthetic cells, there will be questions from other disciplines, such as philosophy, ethics, medicine even. And what is now a synthetic cell? So a synthetic cell should contain some new functionalities, synthetic modules, but it should still have all the key features of a natural cell, which includes a chemical boundary or a membrane, an information system, which is DNA in a normal cell, a way to convert energy or a metabolism. It should be able to grow and reproduce and adapt to the environment. So what are some possible applications of synthetic cells and why do we try to create them? They can be used in origin of life research, because when you know the conditions you need to assemble complex synthetic cells, you will also learn about which conditions might have existed on Earth at some point to create life. Then they can be applied in biotechnology as nanofactories. For instance, you can create within synthetic cells drugs that would be toxic for natural cells and produce them within the synthetic cell. Then in medicine, they can be used as highly specific drug delivery systems. They can even be applied in biocomputing because they are very easy to manipulate, and even in cell biology to learn something about natural cells. Because when you can mimic the behavior of a natural cell in a minimal manner in a synthetic cell, you will learn a whole lot about natural cells as well. And this research field just started in the last 10 to 20 years, and there will be many more applications when we make more progress. So when I looked at the student questions, there were quite a few questions about if biology and chemistry can be combined to create a synthetic organism. And actually, this has already been done. So researchers in the Craig Venter Institute in the US produced synthetic genomes from bacteria called mycoplasma. And mycoplasma are a good candidate for a minimal life system because they have the smallest known genomes from all organisms that only contain about 1,000 genes, whereas most other bacteria have up to 5,000 genes. And the reason why these genomes are so minimal is because mycoplasma are parasites of eukaryotic cells, so they hijack a lot of the machinery of the eukaryotic cell and thus lost a lot of their genome during evolution. So chemists then synthesized chemically in the lab the genome of Mycoplasma mycoides from the bottom up using a method called Gibson assembly method. So in principle, they synthesized long strands of DNA, DNA oligomers, and then annealed them using PCR technology until they reached very long DNA fragments and in the end, the whole synthetic genome. The next step was then to transplant this genome into another Mycoplasma strain, Mycoplasma capricolum, which that had its DNA removed. So with that, they created a new organism that they called Mycoplasma laboratorium, or JCVI Syn 1.0. In subsequent work, they could reduce the genome further by half so that there were only 500,000 base pairs left. And this new organism is now the third version of their first Mycoplasma laboratorium. And this minimal organism only has around 440 cells. And very interestingly, even in this very small genome, around 20% of all genes don't have an assigned function. So there's a lot to discover even in such simple organisms. The organism also contains about 35 RNA molecules. And this work defined the minimal set of genes required for life. And it also tells us a lot about synthetic cells. So that's the minimal complexity we want to achieve someday to produce a synthetic cell from the bottom up. However, this research did not truly use a bottom-up approach. So they first produced a synthetic genome from the bottom up by chemical synthesis. However, then they used the host cell, whose DNA has been removed, and then just introduced the genome into the host cell. But if we want to truly produce a synthetic cell from the bottom up, we have to start with individual modules. So first of all, we need a membrane. Otherwise, we cannot contain and concentrate all the biomolecules to high enough levels to produce chemical reactions. Then we need an information system, which is DNA normal cells, but there are artificial ways to do that in synthetic cells. Then we would need molecular machines to translate the information stored in the genome to something useful. We need an energy source, and only then we will have a true synthetic cell. So at the moment, we try to assemble all these individual modules, and in the future, we hope to bring them all together to create a synthetic cell. Now I will talk a little bit about my research, which mainly is focused on the membrane part and the energy part of synthetic cell. I already talked a little bit about synthetic genomes, but I won't talk about molecular machines today too much. So how can we produce a synthetic vesicles that can be the container for a synthetic cell? For that, we should first look at the membrane of a natural cell. So membranes are assemblies of phospholipid molecules, and a phospholipid is very similar to the detergent molecules we have seen this morning that are used in soap to fight viruses. However, they have a slightly different geometry. So they have two hydrophobic tails and then the hydrophilic head group. And because of that, they self-assemble in a tail-to-tail manner into phospholipid bilayers. And if you continue this phospholipid bilayer, it will actually bend into a sphere. And this is exactly what we see as a cell membrane. So it's a layer of phospholipids. So the question is now, can we produce synthetic vesicles that resemble cells in the lab? And actually, it is not that hard to do. We can dry down lipid films, rehydrate them, then in buffer in an electrical field, and they will spontaneously assemble into these giant vesicles, also called liposomes. Here you can see a fluorescent microscopy image of such vesicles, and they just look like cells whose membranes have been labeled with a fluorescent molecule. So interestingly, this process can be streamlined using modern engineering techniques, so microfluidics. When you have an oil phase with all your lipids and then add water droplets in a very controlled manner, such vesicles will be formed spontaneously, and with microfluidic technology, you can control this process and produce them in a streamlined fashion. So interestingly, you can not only use natural lipids, but you can modify your lipids and use synthetic lipids to give the membrane some different properties that don't exist in nature. And also importantly, you can adjust the size of these vesicles. So you can produce anything between 20 nanometers and up to 200 micrometers. So from small virus-like sizes to cell sizes or the size of a big eukaryotic cell. And these are really the vesicles of choice to create synthetic cells. However, they are not that stable and not that easy to create in the lab. And this is where chemistry can be very important. So chemists try to mimic the behavior of lipids by synthesizing molecules called block copolymers. And as lipids, they consist of a hydrophobic tail and a hydrophilic head group. They also self-assemble when rehydrated. However, they are chemically much more flexible, so you can change the functional groups of these polymers. And they also have been shown to be much more stable than liposomes. For instance, one copolymer that is used quite often is called PDMS-GPEO, which is a polydimethylsiloxane that makes up the hydrophobic part of the molecule, and then the PEO, the polyethylene oxide, is the hydrophilic part of this amphiphile. So with that, I have shown you how to produce the membrane. And now the next module, and this is where my main research focus is how to convert energy in such synthetic cells. And for that, let's have a quick look at how animals produce or convert their energy. So when we take up nutrients with our food, we will take up sugars, fatty acids, and amino acids, and the metabolism then transfers electrons from these processes into mitochondria, into the citric acid cycle, where reduction equivalents are produced, and these electrons are then fed into a process called oxidative phosphorylation, or the electron transport chain, and there the electrons are then converted into ATP. And ATP is the energy currency of cells and then drives all other biochemical reactions within the cell. So if we want to produce synthetic cells, it will be very crucial to produce ATP. A human produces about 50 to 100 kilograms of ATP per day. So you basically produce your own body weight in ATP. However, you won't gain any mass by that. It is recycled so quickly. So in theory, every ATP molecule is used up to 1,000 times per day. So is it now possible to reproduce this process in a test tube? For that, let's have a closer look into oxidative phosphorylation. So during oxidative phosphorylation, electrons end up in the electron transfer chain, which is a series of proteins that are able to transfer electrons. The initial protein is called complex 1, and this is the main research focus of our group. So our group looks at the structure and mechanism of this complex, but also in its involvement in diseases and how we can produce new therapeutics to overcome diseases involving that complex. I am mainly interested in using it in synthetic biology applications. So I try to approach the research in our lab from a bit a different angle. And electrons are now fed in to the system from the citric acid cycle, are then transferred to quinones, which we have already seen this morning when we talked about batteries. And during that process, protons are transferred across the membrane. So they are concentrating on one side of the membrane, and with that you build an electrochemical potential. The electrons are then transferred through complex 3 and 4, and further protons are pumped across the membrane. Complex 4 is very interesting. It is the last protein of this electron transfer chain, and it reduces oxygen to water in a highly efficient manner. And this is actually the reason why we all breathe oxygen. It's because of this enzyme. The electrochemical gradient that is produced during this process is called proton motive force and is then converted by ATP synthase into ATP. And as an analogy, you can think of a power station. So the proton motive force would be your dam that stores the energy, and the ATP synthase is your turbine that just converts it into an energy form that is usable by the cell. So is it now possible to mimic this process in a test tube? The problem is that when you purify these proteins, you have to add soaps again, detergents, because these proteins are very hydrophobic, and if you just use aqueous buffers, they would crash out and lose their structure and activity. So you have to add detergents to purify these proteins. And with that, you also destroy the membrane. Thus, you cannot produce an electrochemical proton gradient. So if you try to purify them and measure their activities, you have to bring them back into a liposome. And this you can do by actually removing the detergents and mixing the different proteins with the liposomes. And my research focuses on developing protocols to achieve that. And I want to use very small liposomes that are only around 50 nanometers in diameter, because then I can insert them later into synthetic cells, which are much larger, which are in the micrometer range. So what I have been doing, I produced a synthetic organelle that is able to drive ATP synthesis. So this is like a minimal oxidative phosphorylation chain. You start with complex 1. So I have isolated complex 1 from cow hearts. And when you add now NADH to this system, so that's the electron donor, protons will pump into the vesicle. The electrons end up on quinone. The quinone will be recycled by a complex 4 protein that I isolated from E. coli. And then the proton motive force is then used by the ATP synthase to produce ATP. So here you see just some raw data, an actual experiment. When I add NADH, I can see that ATP is produced over time until I add an uncoupler. And actually this system, even though it is quite simple, it can produce ATP continuously over several hours, and thus it can be used in synthetic cells. So once you can produce ATP in synthetic cells, you can control a lot of different processes, such as locomotion, active transport, cytoskeleton synthesis, macromolecule synthesis, as well as signal transduction. And at the moment, I'm working on incorporating those synthetic organelles into synthetic cells to drive then such other processes. With that, I'm already at the end of this part, and there has been great progress in assembling modules of synthetic cells. However, a true synthetic cell so far has not been created from the bottom up. Chemistry will be very important for the future of this research field because it will allow to introduce new functionalities into synthetic cells, which is very hard to do in natural cells. And also synthetic cells have many applications and have the potential to revolutionize different fields of science, such as origin of life research, which will be introduced by Claudia after this talk. Thank you very much.
Hi, everyone. Thanks a lot, Oliver, for a very nice talk and also for introducing the topic of my talk. So I will now share the screen with you, and I'll use the same kind of slides that Jenny used this morning, going back to your big questions. And it's very nice to see that quite a lot of you were interested in understanding how chemistry can help us find out and explain the origin of life. And the reason why I'm very happy about this is because that's what I've been doing for all of my research career, from the PhD through the postdoc. So we really want to think about is that if we want chemistry to explain or to tell us something about the origin of life, what we need to think about is what we are referring to. So if we imagine to shrink the whole life of Earth in one day, you can see here that we would just appear around midnight, and it takes quite a lot of going back in time, about 4.5 billion years ago, to have the formation of the Earth. And about 500 million years after that, more or less, we would imagine to have the origin of life. And so you can imagine that when we talk about the origin of life, we're not talking about the kind of life that we have nowadays on our planet, but still we are thinking about something that we would consider alive. So what we need to think about when we try to understand how life started on our planet is how our planet was about 4 billion years ago. And the answer is that the planet was very, very, very different from the planet that we have today. The first thing that you can think about, for example, is that now we have oxygen in the atmosphere, and we know that oxygen is fundamental for life. But back then, 4 billion years ago, there was no oxygen in the atmosphere. And so when you think about even just these small things, you think about how difficult it might be for us as chemists to understand how the building blocks of life could have formed on early Earth, and then how they could have assembled to form the cells that Oliver was just showing you. So what kind of environment, what kind of planet was Earth 4 billion years ago? Well, what we know is what we learn in geology. So geologists can give us information about the crust, about the Earth's surface, and astronomers can give us information about the atmosphere, about, for example, what kind of gases were available on early Earth on the atmosphere, or how much sun would be able to heat the surface. Because something else you might think about is that now we have the ozone layer around the Earth. It wasn't there 4 billion years ago. So the amount of light that we would get from the sun was much higher on early Earth compared to what we have today. And so what we know about what was present on early Earth 4 billion years ago is that there were some very small molecules that today we consider to be very toxic. One of them is hydrogen cyanide. So we are very aware that hydrogen cyanide is a very toxic molecule. But what we've been finding in the lab, is that from hydrogen cyanide, you can build, for example, nucleotides. These are the molecules that I've shown you this morning when we were talking about the viruses. And these are the building blocks of RNA and DNA, so of the genome that Oliver was mentioning before. From hydrogen cyanide, you can also form the amino acids. So all of these acronyms that you can see here are some of the 3-letter codes that we have for amino acids. And you can see that quite a lot of them would just derive from hydrogen cyanide. And also, Oliver has talked about membranes and lipids that you need to make membranes. And we talked about them this morning as well when we were talking about the lipid membrane of viruses and soap. And so from hydrogen cyanide, you can also form lipids. So this is just a very complex slide that is intended as complex to give you the understanding that it is difficult to build the building blocks of life. It is difficult to understand exactly what happened to build the building blocks of life. But what we know is that right now we have different pathways that start from just one molecule that we know was available on early Earth. And from that one molecule, using UV light, for example, and other kind of molecules that were available, we can build all the building blocks of life. So Oliver has talked about how a modern cell is made and how you can make one synthetically. And he said very correctly that if we think about a schematic version of a cell would be made of genome, which contains the genetic information, metabolism, something that drives all the activities inside the cell, and membranes, which contains the genome and the metabolism part, so contains everything that is inside the cell to make it an entity. So what kind of cell would have been forming from the building blocks that I've just shown you before on early Earth? Well, if we think about a schematic version of a primitive cell, then we would imagine to have something very similar, just much more simplified. So we would have a primitive membrane, or also called protomembrane, a primitive version of the genome, protogenome, and a primitive version of metabolic systems, protometabolism. So I'll just show you very briefly something that has been done in this past 20 years or so on each of these specific components of early cells. So if we start from genomes, what we know is that in order to form nucleic acids, what you need to have are, so we know that our DNA is organized in a double helix. So there are 2 DNA filaments that are attached together and that form a helix. So if you want to imagine how this could have happened on early Earth, how you can form such a complex structure, what you need to think about is that on early Earth, you would have probably had the monomers. So all of those molecules that I've shown you before, the nucleotides, need to be connected and bound together in order to form a filament. And then this filament would need to be interacting with a second filament so that it could form a double strand and then forming the helix. And so what we need to understand as chemists or as biochemists is how this can happen, how we can bind together all the monomers. And this is just to give you an idea of what we need to do. So what you need to have is you need to have a nucleotide, so a monomer, which is activated. So it needs more energy, it needs to be active to be then bound to the rest of the filament and keep elongating the filament that you are building. And then, of course, after you imagine to have this double strand form, I have here a video that I hope it will show just to give you an idea of what I'm talking about. So this is a movie that was made to explain the process that I've just described you. Dark blue is the filament that is already done. And on top of that, there's a light blue filament that is forming by joining together all the monomers. So I hope this is running. This is just to show you how we need to imagine these things happening and then how we can study them. And after you've made this double filament, what you need to think about is how you can keep cycling this so that you open the filament and you can build more so that you can replicate the DNA. If we just go briefly on the metabolism part, what we need to think about is, as Oliver was saying, these kind of systems are very complex. So this is just a very simple schematic representation of the electron transport chain. But of course, such a complex system would have probably not existed on early Earth. So what we need to find out is whether something much, much simpler could have existed on early Earth and could have formed and behaved in the same way of the complex machineries that we have today. So this is just to show you that there has been research done in this direction as well. And you can see here that there's has been research on mimicking this electron transport chain, which is and in mimicking it in a much simplified way. And in the right figures, microscope figures that you can see here, you can see that the iron-based mimicking system is attached to the membrane. And this graph just shows you that if you look at the light purple and dark purple bars, you can see that there is a way to discriminate some mimicking systems from others that don't work. And then I'm just not going to talk that much about membranes because Oliver has already talked a lot about that. But the question I want to address is what kind of lipids would have been available on early Earth? Well, we know that lipids were likely very available on early Earth. It's just that they were not the same kind of lipids that are available now. So on the left, you can see the kind of lipids that would have been available on early Earth. On the right, the lipids that form our membranes today. You can see that there's a very big difference between the two. On the right, much shorter chains. So the gray part is much shorter compared to the right lipid. And the head group, you can see that on the left, we have only oxygens in the head group, and on the right, we have phosphate groups. So the question that we've been asking is how you can go from one to the other. And the answer we've been giving, and I'm not going too much into details in this, is we can synthesize chemically something that has an intermediate structure. And we did that, and these are just some examples of the same vesicles, similar vesicles, and similar membranes those that Oliver showed you before, just to show you that we are doing something about every bit of the components that make a primitive cell. And the open question that I hope that I want to ask you, and that I hope some of you will answer during and after your degree in Cambridge, is what can we make these primitive cells do? Can we show that these primitive cells can actually behave in the same way as modern cells? And so this is just to show you that this is the kind of primitive cells we are thinking about, but of course the complexity of modern cells is much, much higher. And so there's a lot to do, and I hope you will fill the gap between the primitive cell on the left and the modern cell on the right. And with this, I'll give back the word to Jenny.
Okay, thank you very much, Claudia and Oliver. Could I get Oliver to come back on as well? So thank you. That was so fascinating to learn about both of your research and for answering our big questions. So I wonder whether we can continue with answering some of the pre-submitted big questions. One of them, and I think I saw that being asked in the Q&A as well, can inorganic life exist? Who would like to start?
Maybe I can start.
Yeah, Oliver, thanks.
So if you define organic life as carbon-based life, I don't think that inorganic life can exist. There has actually been done quite a lot of research on it. And it has been shown that silicon-based chemistry, although it reaches some complexity, it can never reach the complexity of carbon-based research. And I don't think it does exist at the moment.
Okay, yeah. Claudia, do you want to add to that?
Yeah, so I think what you can think about is what we think life is. And we don't have an answer for this question, and I don't have an answer for this question. But what we can think about is in the way we see life on our planet. I totally agree with Oliver. So if we want to reproduce the life that we have on our planet, it's probably not possible to have inorganic life. But at the same time, what we need to think about is that on other planets, the environment might be very different, the atmosphere might be very different. And this means that alternative situations, alternative membranes, for example, could exist. And I'm just going to give you a very short example. There are some moons on Jupiter where they found that the environment is very different, the atmosphere is very different to the one we have. Same goes for Saturn, for example. And there has been some research to make membranes, so the ones we've already talked about, in solvents that are not water. And what you can see is that you can, if you take the same lipids that we've shown you in our slides, and you put them in an apolar solvent, let's say methane, or because you have to imagine that the temperature was much lower, so it would be liquid, or other kind of apolar solvents, membranes would still form. The only difference would be that the hydrophobic part would now be exposed to the hydrophobic environment, and the hydrophilic part would be shielded. So we can imagine alternative systems, alternative complex machineries working in different environments, but it wouldn't be the life that we are used to talk about.
Wow, that's fascinating. Okay, well, let's move along, just in the interest of time. There's another question that was pre-submitted. Is there a limit to the number of unique compounds, combinations of elements that can be created? Oliver, do you want to start again?
Yes, I can do. I actually investigated a bit in this. And if you try to build a molecule that consists only of 30 atoms, you can use only functional groups that we know of at the moment. You can already produce 10 to the power of 24 different molecules. And even if you then go more complex and, for instance, want to build something that is 500 Daltons, you can already produce 10 to the power of 60 molecules. So there is a limited number of molecules we can create. However, it's far beyond anything we can test in the lab.
Okay.
So we have, as chemists, endless possibilities.
Great. And Claudia, did you want to add to that at all?
No, I totally agree with Oliver. I think we have endless possibilities, and right now the limit is just the technology that we might have, either to build them or to test them.
Yeah, that's really exciting to know. Okay, so maybe I'll end with one question before I pass it to Naomi to ask the rest of the questions, because I can see the Q&A questions building up to do with your research talks. So why is it for you chemistry is the best science? And I know that both of you work on the boundaries of biology and chemistry. So maybe you can comment a little bit about that. Oliver, would you like to start?
Yes. So for me, chemistry is not the best science. But I like to combine chemistry with biology. And I think the best science nowadays is interdisciplinary. And chemistry is the foundation of a lot of future interesting research projects. And I think that chemistry has to be applied to more difficult, more complex problems, and you have to work with other scientists to really do the science that is needed in the 21st century. So I really enjoy chemistry, but I think it should not be used on its own. It should be combined with other subjects.
Yes, very important. And Claudia?
Yeah, so I'm just going to say that I'm going to ask you, Jenny, the same question right after I reply, so just be prepared. So I think I'm going to combine in some ways the answers that Alex and Oliver have been giving. So the reason why I think chemistry is the best science is because it's completely interdisciplinary. So if you study chemistry, you can go in such a huge amount of different directions. You can work at the interface with physics, you can work at the interface with maths, you can work at the interface with engineering, biology, even philosophy or ethics, as Oliver was saying. So that's one thing I love about chemistry. Another thing I love about chemistry that Alex talked this morning about is the fact that you can actually do something practical. And I love being able to do something, see something with my own eyes. And I think that's amazing. And the third reason is that chemistry is not something that you do on your own, but you actually work in a group, you work with people, you interact with people much more than what you would do, for example, in other disciplines in which, for example, you work with your computer or with books or things like that. Not that those other disciplines are not engaging or to be done in a community, but I really enjoy the community part and the social aspects of chemistry.
Yeah, fantastic.
So
yes, from my side, I completely agree with all of the answers, and I loved hearing about your answers. So for me, chemistry is the world. It touches on every aspect of our lives, from the food we eat to our DNA to the air that we breathe, the medicine that we take. Yeah, so it's highly practical, but at the same time, you can use it as a tool to study really important questions, which we hopefully illustrated this morning. And solve problems. And it's really unique as a science, in my view, because it is the central science, and it's one of the sciences in which you can use to connect to other fields and other sciences. So it very strongly connects to biology, as we saw this afternoon, but also to physics, to material science, and also to the applied sciences. So engineering, medical sciences, you need chemistry, a good foundation in chemistry to be good at all of these. So that's why I think it is the best science, in my view, obviously, but everyone's view is correct. Okay, so I think that brings us nicely to an end. And Naomi, I'll let you take over with the question asking, because I think I could see a lot, and you probably had a better chance of filtering.
Sure. So we've probably got time just for maybe 2 or 3 questions, I think, at the end now, because then people might need to go to the end of their school day. First of all, just to say thank you so much, all 3 of you, for your contributions there, for the questions, and really great to hear your view as well on chemistry and biology and why you do the sciences you do. I think that's really inspiring for our attendees to hear. So thank you for that. So just going to pick out a few questions here relating to kind of your research talks. So first question I'm going to ask, has there been research into creating synthetic cells which could potentially survive and adapt to future changes to the Earth, say in 100 years?
At the moment, this has not been achieved yet, but there are groups working on that, and it is for sure the future of the field. But at the moment, we are still very limited with the creation of a complete synthetic cell. So we are very good in assembling all the individual modules, and we now try to combine different modules, but we are still not there in producing a full synthetic cells. But I think this will be achieved in the next 20 to 30 years.
Thank you. I think the next question is probably for Claudia, more related to your research talk. So could organic species like plants have consciousness, even if it's not in the same way as us?
Wow. That's such a profound question. It's an amazing question to which I don't have an answer. You know, I was mentioning before that we don't even have a definition for life. So this question would bring us to a definition of consciousness. So, you know, it's very, very complex. I think we are, what we are aiming to do is to understand how, using the knowledge that we have about early Earth, for example, to understand how the chemicals that were available there, the molecules that were available there, could build up into something more complex. And now we have my cat that is going to join the conversation. But it's very difficult to know how far we are going to go in that direction, because of course we cannot go back in time. I don't know if Oliver wants to say anything about this question, because you also raised the point about ethics. So maybe that's also something you can.
I think that's something that is very difficult to answer at the moment. There we really have to involve philosophies that have to define what is a conscience. And at the moment, I don't think we can say that. Yeah.
A very big question, but it's great to see our potential students engaging with really big questions like that. And I'm sure you'll reach far with thinking about big questions like that. I'm going to make this the last question of the day, because I think people probably need to go to the end of their school days. So what is so special about carbon that makes it good for forming the structure of life? And actually, we've had quite a few questions about carbon come in throughout the afternoon about sort of why carbon and what makes that so great.
Who wants to start?
So something I can say is that so clearly carbon is an amazing element. You all have studied carbon in different ways and different topics throughout the years you spent at school. So you know that every time you touch a new topic, carbon is going to be there in some way. So it's something you are used to think about already. And what I can say is that for what concern origin of life, carbon-containing molecules were very available. So one thing that one might want to consider is the availability and the abundance of a certain element. Because if you think about, you know, we've mentioned before silicon. If we think about the abundance, it's very immediate to see which one of them is more available, would have been more available on Earth. Carbon has the ability to bind a huge number of different elements, form molecules with different geometries. Not all the atoms, so not all the elements can do the same. So the properties that carbon has for its position on the periodic table and for its size and shape and everything makes it, I think, special. I'm sure that Jenny or Oliver might want to add up on that.
Yes, I completely agree with Claudia. It's mainly the chemical properties of carbon that make it so interesting, and it can really go. So proteins, for instance, are mainly carbon-based. DNAs, RNA, so all complex molecules, biomolecules, we know are carbon-based, and carbon can undergo up to 4 bonds with different other elements, and this makes it very special, and it's also very stable.